This is the first webinar series of the Mod Alliance. The Mod Alliance is a collaborative platform comprising of communications efforts of 3 Horizon Europe sister projects, which is MARCO-BOLO, that's the M, Obama Next, that's the O, and DiverSea, that's the D. The projects in the Mod Alliance are funded under the same Horizon Europe call topic, with a shared focus on observing and mapping biodiversity and ecosystems, with a particular focus on coastal and marine ecosystems. The Alliance is aiming to unite our efforts and amplify the impact of each one of our projects. We're seeking to share progress, promote collaboration, and advocate for the effective conservation and management of coastal marine environments. The 3 projects will work to complement each other towards common goals through joint initiatives like this webinar series and dedicated communication web pages and outputs. And we're striving for positive change in marine management, policy, and conservation. This is the first of our webinars, and the first webinar is hosted by DiverSea, the project I'm coordinating. The second will be moderated by MARCO-BOLO in the beginning of 2025, and the third moderated by Obama Next after that. It's with great pleasure that I introduce our presenter for today, who's Dr. Raes. He's a marine ecologist who works on showcasing how environmental DNA and genomic tools can be scaled and integrated into the day-to-day operations of conservation and fisheries managers to enable rapid and robust monitoring in ocean health. Utilizing a multidisciplinary research approach, he synthesizes data into practical information, bridging fields like microbiology, biogeochemistry, and marine ecology in coastal and open oceanic environments ranging from tropical to polar ecosystems. Today, the seminar presentation is entitled The OceanOmics Program, Monitoring and Protecting Ocean Life. The OceanOmics program is a Minderoo Foundation initiative, and its primary goal is to safeguard marine biodiversity by developing and scaling genomics techniques for monitoring marine wildlife. The program strategy centers on harnessing cutting-edge genomic techniques and multidisciplinary expertise to generate high-quality ocean-scale data, helping to understand marine species distribution and range shifts, and informing effective policies and management strategies. There'll be questions after the presentation, and the way we'll manage them is feel free to ask questions during the presentation in the chat. Please leave your name and the question. I will collate those, and at the end of the presentation, I will call on the question asker to ask their question themselves, or if they don't wish to, because they don't want to be recorded, please let me know in the chat question and I will ask the question on your behalf. So with that, I give you Eric for the presentation.
Share my presentation as a start. Thank you very much, Glenn, for the introduction. Sharing it and hoping that you can all see my first page.
That's great, thanks.
Wonderful. Hi, everyone. Hope you're well and having a good week. My name is Eric Raes. I work at the Minderoo Foundation based in Perth, Australia. The Minderoo Foundation is a philanthropic organization founded by Andrew and Nicola Forrest back in 2001. The vision of our foundation is a society that values all people and natural ecosystem, and the mission is to forge a fairer future. I would like to start by acknowledging that I'm giving this presentation on Wajak Kujar country, or Perth, and that the Noongar people are the traditional owners of this land and also the custodians and the first marine scientists and inventors of the country for which I'm giving this presentation. Big shout out to Glenn, and thank you very much for the Mod Alliance for the opportunity to present our team's research here today. So in our team, I'm a researcher in the OceanOmics team, where we're using environmental, eDNA, marine genomics as non-invasive, rapid, and scalable tools to monitor marine wildlife. In our group, we pretty much work across the marine genomic workflow with our colleagues working in the molecular innovation space, where they're looking into eDNA-based forensic approaches to detect, classify, enrich, or deplete species, populations, and individuals from seawater. Our computational biology and AI team are really looking into how we can transform the way that we extract information from eDNA, including novel AI tools and data visualization. Colleagues from the Ocean Genome team are filling in the needed gaps of the reference genome libraries that we so desperately need to classify our DNA sequences, with a focus on marine vertebrates, families, and threatened species. And our scaling at sea efforts, close to my heart, is where we focus on the development of ship-based and autonomous, semi-autonomous platforms that will enable the ecosystem scale deployment of eDNA. Across this workflow, we focus on impact projects that really look into the translation and the application of eDNA. We have a variety of partners, including the Australian government, Parks Australia, different musea that really help to hone down into these ocean genomes and tissue samples, industry, a number of different institutions. We have a dedicated marine genomic research facility at the University of Western Australia, based in Perth, where we can process samples, eDNA, tissue samples. We both have short and long read sequencing technologies, computational biology resources, all based at the laboratory university. One of the projects that I would like to highlight is a 3-year partnership that I'm leading with Parks Australia, which is focusing on how we can provide a proof of concept how eDNA can be deployed as a cost-effective way to monitor biodiversity in Australia's vast marine parks. A big focus as well is the translation to policy, which Glenn already mentioned, and it's kind of helping to define or steer the way with our partner how eDNA can be implemented into management, policy, and regulatory frameworks. Another aspect of this partnership is providing evidence-based science to how we can standardize eDNA as a biomonitoring tool. And we're operationalizing that through dedicated research expeditions shown here in this figure, going from the tropics, Cocos and Christmas Island, all the way to the temperate regions, up to the Southern Ocean. The different colors that denote different expeditions that we've conducted to date, 15 so far. And the circles are surface eDNA samples, and the squares, the pink squares, are deep sea eDNA samples, which we collected in collaboration with our partner based at the University of Western Australia as well. These are the deep sea landers that are deployed. They have beta traps here, cameras, and we're collecting eDNA samples from these deep sea environments as well. The marine parks that you see in this figure are shown by the green polygons. I'm going to go a little bit into detail in this kind of map of the expeditions that we have so far, as the scale that we're talking about here, all the way from the north, from Christmas and Cocos Island, down to Tasmania, is around 6,000 kilometers. If you go from the northern top of Norway all the way down to Faro in Portugal, we're talking about 4,000 kilometers. So it's huge scales where we think that eDNA can be used as a tool to kind of like assess vast spatial areas, but also temporal scale. Here in the Abrolhos Islands, where we zoom in, sit nicely at a transition zone between tropical water that are coming from Indonesia, coming down with the Luwang current, and colder waters from the Southern Ocean. And you get a really rich biodiversity hotspot over here. We've done 4 annual surveys here. Next year will be the 5th. We're working closely together with the local custodians there, the Yamaji people. Sea rangers come on board with our expedition. We have a nice 2-way knowledge exchange. So it's a combination of large spatial scales that we're trying to tackle and temporal variation. These islands are really prone to marine heat waves, which are accelerated when we have a La Nina year, which is happening at the moment. And so a lot of data, but I will jump into some more of these. I have an issue here with the band to see himself rather than the slides. Anyway, in 2022, we did an expedition from Perth all the way to Cocos Island, which you can see in the top right-hand corner. And our question was kind of whether we could pick up DNA traces from pelagic fish and whether we could identify known latitudinal variation. This image here on the bottom shows Perth and our sites that we sampled all the way to Cocos Island. And at each site, we collected 5 samples, 2 liters, filtered through a filter membrane, 0.45 micron. And the size of the bubble indicates how often a certain family, which you see here on the y-axis, was present in our replicates. If the family was present in 5 of our replicates, large bubble. If there was only present in 1 out of our 5 replicates, we didn't even bother showing the presence of that certain fish family. So what we see is kind of like what we expect, temperate species close to Perth, and then we kind of see like a nice transition to more tropical fish when we cross the Tropic of Capricorn. In the tropics, we see these flying fish, Exosuridae, nicely pop up in our like data set. We have these mesophotic deep sea cryptic species as well that pop in the data, like these Stomidae that we kind of see occurrences here, and as well the Myctophidae, kind of these little lanternfish that are like prevalent pretty much throughout the data. It's a nice figure of kind of like synthesizing latitudinal variation across scales. But what I want to delve into a little bit more is kind of like the power that eDNA have, or the potential to contribute to global observation data. This figure here on the top right is showing observational data from OBIS, species marine fish sightings. And what we see is kind of like gaps and higher concentrations of observations, obviously near the coast and the continental shelf, but a paucity of data in the Eastern Indian Ocean, a lack of data. Now, these 2 figures here on the bottom are showing observational data from OBIS in the light blue and our eDNA presence data in the dark blue. We recorded the dark blue, a sighting or as a unique eDNA detection that appears at least in 1 replicate in our sites. So even if a detection was present in 4 out of our 5 replicates in 1 site, we just consider this still as 1 presence, 1 sighting as a site. And what is really nice is that we see kind of a promising kind of the promising power of eDNA. They're actually contributing to observations that were not present in OBIS. 1 cross 2 different mitochondrial amplicon assays. One that really stands out here are the Giganturidae, or the telescope fish. In this region, in the Eastern Indian Ocean, we only, in OBIS database, only 5 sightings were recorded, but with eDNA data, these are not relative abundances, but actually presence data in the replicates per site, we recorded 55 sightings. So it's really showing that the power of eDNA can contribute to global observation and even fill in some of these gaps in the Eastern Indian Ocean here. So on the y-axis, we have a variety of different fish families, where we see that eDNA data in dark blue are giving a significant contribution. Another example from that similar data set, nicely illustrated by this color image from Global Fish Watch, which illustrates the intensity of high seas fishing, fringing the exclusive economic zone of Cocos and Keeling Island, Christmas Island, where we see the presence of fishing vessels which have their automated identification system turned on by the little green dots. And so what we wanted to ask is like whether we could correlate fishing pressure with eDNA. To test that, we did a transect to Cocos Island in and outside the exclusive economic zone, which are color-coded, and specifically look into a commercial fish family, the Scombridae, which present tunas, mackerels, bonito. And while we do not see a significant difference between inside and outside the EEZ, which is also what we can expect from this little snapshot, and because the Scombridae are also a highly migratory species, what is really neat to see in this data is that at every site we were able to detect the presence of Scombridae. Again, like opening up like eDNA as a tool or as a triage tool for monitoring for commercial fish species, in addition to other tools, of course. Similarly here, the presence, we got a really high reed abundance for this marlin, and that was exactly at the same spot where we have a really enormous seamount, which rose up from 5,000 meters to 16 meters below the surface. And now this whole exclusive economic zone has become a marine park. And so the idea here is that when you start to protect habitat, you will also start to protect marine species. And eDNA is again a tool that you can deploy at vast spatial scales here to detect these iconic species, whether they are present with the measurements that you put in place, such as a marine protected park. When we go to these islands, they're called Indian Ocean territories, both Cocos and Christmas Island. We make sure that we engage with the local community. We go to the schools. We engage in ocean advocacy, ocean literacy, because these people are our next ocean advocates for the future. It's very rewarding, and it gives a totally different scope as well compared to the pure science that we're doing. It's engaging a lot with the marine park rangers on the ground, but also with the fishery rangers who have a great understanding of the ecosystem that is happening. And they're also interested because we come onto their country and ask, what are you actually doing with all of this eDNA data? But it's also a good way to ask, what do you want from our eDNA data? And how can we help visualize certain aspects or species of interest that you want to see? And at the end of the year in November, next month, we're going back to both Cocos and Christmas Island to kind of like sit down with the community and the fishery rangers and to show these kind of like visualizations. They asked us to show this is an image from Cocos Island and interested where we detected the Maori Raes. This is like a species of interest for the local community. The blue were sample sites from 2023 and the orange from 2022. The crosses were also sites where we collected eDNA, but where we didn't had a detection of the Raes. Similarly, around Christmas Island, the local fisheries, they were interested where we would find the wahoo, a very recreational species. And it's kind of the combination of using these visualization tools that we bring back to the community who specifically asked us of species of interest. Then we can sit down with them and go through what kind of how eDNA can inform us on trends on habitat use, species distribution, and potential temporal shifts and changes over time. And it's a two-way communication and also that knowledge transfer, even digging down into which kind of different assays are more specific or give you different results for a species of interest. For example, these two figures, again, show how many times a wahoo popped up in our 5 replicates around Christmas Island. But there's also some, of course, some specificity. The 16S showed us like a higher presence across the different replicates, and bringing this information back to the locals. It's really helpful because next time when we go out, they will also like take us around the island and show us which sites need protection and where we should potentially put boundaries and protection measurements around it. So engagement with local community, very important. Also for us to help, how do we visualize our like data? Another example which I wanted to dig into now across different spatial scale, rather small resolution, is work that we did around the Rowley Shoals. It's in the northwestern tip of Australia, and there are a couple of island atolls where we sampled eDNA on the slope of these reefs and in the lagoon. And what nicely came out, not very surprising, but is that eDNA was able to pick up fish community differences based on habitats that nicely separated on the habitat use, either the slope or the lagoon. And so these fine scale spatial differences that eDNA is able to answer, now we're talking about a resolution of 60 meters compared to hundreds of kilometers in the Indian Ocean around Cocos and Christmas Island, really like allow us to test these questions. Where do we place MPA boundaries around a marine protected area? And then also further dig in what the fish community differences are between the lagoon and the slope using different effect sizes. So we have more Pomacentridae in like the lagoon and more Gobiidae. So this again, it's like a really small spatial scale, but it's not only the aerial size of the marine protected area that is important. It's also important to know the diversity that we have within a marine protected area. And that's another thing that eDNA is able to answer. So this figure here shows the different ASVs or haplotypes for one specific fish on the narrow banded silver side. The colors stand for these different atolls that we visited. It even shows a little bit of like endemism, endemic haplotypic variations from a little tiny fish. All the red ones here are coming from Montebello Island. And I think it is very interesting that we can go from large spatial scales, finer spatial scales, to the genetic variation within these environments. And that genetic variation is obviously different for different fish species, but it's indeed handy that eDNA is able to give you insights into this ASV or haplotypic variation. Another expedition that we did was along the southwest coast of Australia. Now we're in a mix between states and Commonwealth waters, 3 to 12 nautical miles. But we did this the first time that we did the survey, around 1,000 kilometers long. We did this in 10 days. Impossible to do with divers or baited underwater videos. Again, highlighting the scale that eDNA can be deployed at as kind of like a triage tool to see where we see biodiversity hotspots. Along that coastline, the state government wants to implement a couple of marine protected areas. And again, here, the figures that people are interested in from a state management perspective are kind of like on the x-axis, we now have longitude. We're going from west to east, that side. Fish family species, what are the presence, the absence of certain families? Are we able to detect hotspots? And that's kind of like the stakeholder engagement that we're having on the ground with local rangers and state and national government. On that same trip, we also did a comparison between our traditional Niskin sampling, and we trialed a semi-high throughput eDNA sampler from Ocean Diagnostics, which is able to filter 2 liters on 0.5 micron filters at depth, around 7 minutes per sample. And so the colors here denote different sampling sites, kind of like showing site fidelity rather than differences between the 2 different methodologies in this PCA plot. Circles Niskin and the triangles, the high throughput sampling device, really opening up the doors for robust, standardized, reproducible sampling at depth rather than taking samples of the Niskin, bringing them into the lab and start to like filter. It has a CTD in here and logs all your metadata, latitude, longitude, depth, filter volume. Very nice tool. So where are we going next? We continue to work closely and collaboratively with the Australian government, Parks Australia, building capacity and doing knowledge transfer into technology with people on the ground. The OceanOmics program was doing a lot of work and feasibility studies. Now we're really looking to kind of like partner-driven approach and to kind of look into how can we scale eDNA, because this is like a multidisciplinary, multi-stakeholder effort to deploy it at these vast scales in Australia that we're talking about. Looking into automation of the field sampling through automated samplers, automating lab workflows, miniaturizing lab workflows, and bioinformatic analyses as well. Big component is AI integration as well to produce reports. That is nice work that our computational biology team are doing. This is our eDNA dashboard, which will be coming online soon, where all our sites where we've collected eDNA will be visible. You can click through them and explore kind of like which species were present. We won't be including IUCN species that are listed as critically endangered, or vulnerable. You can get in contact with us so we can give you like the raw data for that. Raw data will also all be uploaded on SRA and CBI. What is interesting is that we're computational biology put this like chat with our data. It's like a beta test on there as well, which is using a large language model or kind of like a ChatGPT style, where we have like a human interface of interrogating data. And that's really came out from discussions with park managers. They don't want to see an OTU or an ASV table or read abundance, rarefied or not rarefied. They just want to have this kind of like a question, and that's something that is in the making, where we can just ask simple questions. Are there sharks in the data? Or can you list all the sites where a parrotfish was detected and list simultaneously with other fish were like detected? There will be a Python script that will be like rolling out next to it, and then you have your answers on like the left-hand side to that. And that kind of AI summary, those summary reports, is also something they were using to kind of like produce a report after expeditions to give it back to the stakeholders, especially when we're working with indigenous groups on the ground, because we have an obligation of returning knowledge to country. And these kind of reports, these are still like draft ones, listing how many sites, unique ASVs, kind of AI-based summary, what kind of fish were found, what these kind of like fish do or don't do. And this kind of AI is kind of large language model is also integrating different data sources. For example, Aquamaps, which is a fish distribution, which shows a fish distribution probability. It's integrating data from FishBase to show which fish species are native to which country. The large language model is also then incorporating the Australian faunal directory. And all of that is then giving us an idea how we can like divide our ASV sightings from the eDNA into validated species that are highly likely confirmed by all of these databases, plausible species that have that occur in some databases but have a high percentage when you blast them, and kind of like potential close relative, kind of like the third category. And such like a report is kind of is valuable for our stakeholders because then they have something in hand and going like, this is plausible. These are the species that you collected at this site. This is what I found. Everything is available on our GitHub. We have now version 1 of our analytical pipeline. It's the Docker station, so we can have your version control. The large language model is also open source, which you can use kind of like validate different species citing. This is just kind of like a placeholder slide. I can share all my slides as well. But you have this as a reference to go to our GitHub. Another project relating to kind of like scaling our endeavors up is exciting work with the CSIRO, which is Australia's National Science Agency. On the left-hand side, you see kind of all our different expeditions that we have done to date so far, but we want to kind of go to an image that would look more like that. Lines on ships of opportunity. And to start, we have a project that is almost signed where Minderoo is funding the research and development of an eDNA autosampler, which will be integrated in Australia's research vessel, the RV Investigator, for 360 days for the coming 3 years, where water will be taken from the underway flow-through line, and the eDNA sample will take a sample every hour. At a later stage, the different sensors, turbidity, CTD, chlorophyll, will be integrated into this eDNA sampler as well. So the sampler has the opportunity to adaptively sample when the ship is going through a feature of interest, an eddy or an oceanographic front, or even marine parks, as everything will be integrated into the chart and the nautical systems as well. Minderoo is also supporting the second MARCO-BOLO technology and instrumentation workshop in Hobart next year, which is an offshoot of OBON, the Ocean Biomolecular Observing Network, which is a 3-day summit focusing on advancing the development of MARCO-BOLO technology and instrumentation. And lastly, capacity building is really forefront and goes hand in hand for us with kind of like the translation into policy, but especially in remote areas. Christmas Island and both Cocos Island are remote places. And we're setting up an eDNA hub on Christmas Island to operationalize an end-to-end eDNA workflow with support from Parks Australia. We will be lending our eDNA container, as we call it, or eDNA, to Parks Australia for 12 months with a ocean diagnostic sampling device. We will supplying them with an iSeq and additional training. So park rangers on the ground in Christmas Island will be able to start collecting eDNA and have an end-to-end workflow, modular workflow that they can use for marine, but also terrestrial aspects. They have an invasive snake, which is eating an endemic skink. So it's not only marine, but also terrestrial eDNA analysis. OceanOmics is an UN endorsed Ocean Decade project, which sits under the umbrella of OBON, the Ocean Biomolecular Observing Network, hence the title of this seminar, Oceanomics, Monitoring and Protecting Ocean Life. And our mission really resonates with the mission from OBON, which is to transform how we sense, harvest, protect, and manage ocean life, which faces multiple stresses, including pollution, habitat loss, and climate change. And so with that, I would like to give a big thanks and acknowledgement to many, many partners, collaborators, institutions, help, and definitely acknowledge Mindru chairs, Andrew Forrest and Nicola Forrest, for their generous donations. Thank you very much, and looking forward to questions.
Great. Thank you, Eric. I have a few questions, probably can predict. I didn't see anything come up in the chat yet, so I'll just start and see how we go. So I really impressive scale of work that's going on there. Like Australia's a big place, and at a country, it's really quite amazing to see at a country scale. I'm kind of really curious about how you go interfacing with stakeholders that aren't molecular scientists. So and just about you did mention that you talk with, say, local stakeholders about their species of interest, and the ones that you talked about were sort of conservation and fisheries species. But I wondered if you had any interest from local stakeholders on invasive species, or whether that's more of a policy sort of interest. And then secondly, I just wanted you to comment on communicating with policy stakeholders and how you actually, some of the issues you might have with communicating what eDNA data can and can't do.
So our policy engagement framework and how we talk about it is greatly aided by our project that we have with Parks Australia. I'm not quite sure what the equivalent would be in Europe, but it probably would be more at like national scale per country that you would have marine protected agency. So that's for the Commonwealth. That is really helping us to kind of like engage with our stakeholders, but also before they always. So, for example, when we go to Cocos Island, Parks Australia gave us a list of local NGOs that are on the ground. One of them is called Cocos Marine Care. And before we are able to apply for our permits, we have to have that kind of outreach engagement and ask, like, what are the species of interest? How do you see that eDNA data is being used? People can be rather protective as well about sequence data. But it's really that relationship building and trust, that's those two, and over like long time. We held a eDNA for management workshop last year here at the university as well. And there we had a number of different people ranging from the Antarctic division. Kyle is from the Yamaji Corporation from the Abrolhos Islands. Kendra here represented Cocos Marine Care as a voice. We had people from Cocos Marine Care from location dialing in, people from CSRO, the Antarctic Division, Biosecurity, and Defense Force as well. But biosecurity people were in the room, and they have their own agenda, very clear, rather easy because their species of interest are rather clear. we have people from parts of Australia here in the top right and corner, really guiding us as well what their science priorities are. But it takes a long time. It's not a straightforward walk, and it's kind of like communicating managers and science at the same time. And some of the outcomes that we had was data management, visualization, sure. It is really, as you already see, collaboration between managers and scientists, two-way communication for these effective management practices. Because it's what came out of these discussions as well is that we need kind of an education across different tiers, going from rangers on the ground that have finished high school all the way to people in Canberra or Brussels in your ends to make decisions. We might have an understanding about kind of DNA, but not even amplicon assays. And that's definitely a hard one to break. The pitfalls of us scientists saying like what eDNA can't or can do. Relative abundance, everybody wants to know how many fish are there in the ocean, but at the same time, eDNA is giving us more information than baited underwater videos. It's giving us more information than a natural. There are pitfalls with those methods as well. So we have to put them in comparison to the existing methods that we have. So sorry, I'm going to be all over the shop as in answering your question, but it's a hard question to answer first of all. But stakeholder engagement with other groups. On the top of my head, the Yamaji Corporation are sea rangers who are doing course, doing a bachelor, basically in environmental science on their local country. And having kind of them involved in our eDNA work is really paving the way on how they will be able, it's the way on how they can integrate it. And it's kind of like that knowledge transfer. And it's education comes there. Yeah.
Thanks, Eric. I realized that was quite an open-ended question. And so we have a couple of other questions here. Christina Pavlaudi, she's put a question in the chat. Would you like to ask Eric your question, Christina? And then we have Luis Felipe after that.
Sure. Let me open my camera. Hi. Very impressive project. Great talk. I was wondering how difficult was it to incorporate eDNA into the monitoring policies? Because in the EU, it's still not quite there yet. And I'm thinking like if in Australia it found its way, let's say, into management legislation and into official, let's say, document. Yeah.
We're not very much there yet. So the work that we have with Parks Australia was kind of to provide a proof of concept. And there is the US eDNA roadmap that recently came out. We have a similar kind of eDNA roadmap here in Australia. These documents are really kind of pitching the idea of how great eDNA is, but it's not really talking about how we operationalize the collection, the standardization, nor the integration of policy. And that's kind of the project. Our project with Parks Australia is ending next year in June, and now we're like looking into kind of how to operationalize the collection through ships opportunity, certain eDNA hubs in remote areas where the park managers feel empowered to collect and analyze the DNA, and thereby also get like the knowledge and the understanding with local communities as well. Really get into the drill down what eDNA is, but still keep it simple, but also specific about assays, and show them where the fishes are that they want to fish as well. So it's not only protection, it's kind of like that sustainable management approach. And the policy, we recently had like a 2-day workshop with the people from Parks Australia, and it came to the forefront. I mean, there are, as everywhere, people that are pro and the people that are contra. And so that kind of clash of like, how do you integrate eDNA data into policy management? It can even be, as the example that I showed with OBIS, we can like increase our sightings or observations on very robust data. And this does not yet have to say that we're going to go and quantify. We can show relative abundance over seasonal trends and large scale geographical transects. But at this stage, I think even if we have the sightings present in standardized OBIS, like national databases, we're already a long way, because then it will come. Here in Australia, large parts are not even monitored. So at this stage, we're like, any data is data first. But I think I will have to come back to your question maybe next year.
Thank you very much.
Great, thanks. And Luis Felipe, if you'd like to ask your question.
Yeah, thank you. Thank you very much, Eric, for those impressive. And of course, I totally agree with Christina's comments. There's still a long road here in Europe. But anyway, I was wondering about are you considering, I mean, all the domains of life? I mean, prokarya, eukarya, protists, and etc. I mean, because this is one of the biggest added values of eDNA as well. Not only, of course, the fact that you don't need to harvest big animals, but also that you can bring a complete assessment of whole biodiversity.
Absolutely, I completely agree. It's that holistic approach across the marine food web. At Minderu, we really focus on marine vertebrates. My previous work was all on prokaryotic and eukaryotic phytoplankton. And I've been driving that message here at work. The work that we're going to do with the CSIRO on the investigator and the automated sampler there, they're very interested in the bacterial and phytoplankton component. And so we committed to doing 5 amplicons here at university, 2 fish, 1 shark, elasmo assay, and 2 marine mammals. And with the leftover DNA, they will be doing the bacteria 16S ribosomal, and hopefully we can push here to do the invertebrates to CO1. Similarly, around Australia, there are 7 national reference stations around the coast, which are monitored every month. Some of them have decadal data now. And I'm also really trying to advocate that we have all this biobanked eDNA. Can we catalyze or inject funds to do the autotroph levels, because they have already sequenced ribosomal DNA for phytoplankton, but we have the leftover DNA. Let's get some funding so we get some nice temporal data from time series. But yes, hopefully we can do the whole food web.
Yeah, okay, thank you. Because here in France, we started to discuss with the managers, for example, the French Agency of Biodiversity has funded a small project in order to see if we can calculate the pelagic habitat indicators of the MSFD, the European Marine Statutory Framework Directive, with eDNA data of eukaryotes and microorganisms. And and of course, they are considering, of course, doing eDNA for vertebrates as well. But I think it's a sort of separate ways that we really need to try to combine together. So thank you for your answer and good luck for the.
Thank you. Yeah. And that's something, Luis, that we're looking into as well, kind of like metrics, which is not an easy one. In New Zealand, they have a really nice eDNA for fresh waters, and they have 14 different assays, again, from bacteria all the way to fish. In a freshwater system, it's easy to come up with a biodiversity metrics. In the ocean, it's not as straightforward. So, yeah.
Okay, I think that's it for the questions. I have one more, and it's really about this policy interface again, because that's what I'm most interested in. So you touched on before, you talked about how eDNA you get different results from an eDNA assay than you do from a natural or a baited remote camera. And I think what you're touching on there is that all of these methods suffer from imperfect detection. So we have false positives, false negatives, and true positives and true negatives. And I just wondered about how if that level of detail has been sort of talked about in these discussions when it comes to policy, because that's all about how you interpret these data. So, and I'm quite sure you would have that level of detail when you say that there's the pro and the anti, but I just wonder how far that's got up the chain to talk to actual stakeholders and policy. policymakers.
Yeah. So within Park Australia, you have people that also have their career based on baited underwater videos or other methodologies, and they're just kind of like often sometimes a little bit afraid of losing that because there's only a certain amount of pot of funding, and if you start to use something for eDNA and the rest, you're like decreasing potentially their funds. But then you come into the tricky part as you will need. We have been advocating that eDNA is like a triage tool across space and time to then have dedicated research going on. And that could be a dive survey or a trawl. And the false positives, we tried to eliminate. So our dashboard was online, but we took it off again because we're trying to kind of scrutinize a little bit and say any sightings with less than 100 reads, let's just not like show them. If they don't appear in so many like replicates, trying to come up with kind of like standardized way of kind of like already filtering the data before they're going up on the web. That is then also kind of like reproducible through our like GitHub pipeline. What do we see in our water controls, in our bleach controls, in our node template controls? Having kind of these like steps already linking to different databases. And the more information that we provide, the more that we get people on board. On the ground, everybody is really keen to sample the rangers. They're really excited to learn more about the technology. We get a little bit of pushback from higher up, more so as in like, how do we integrate? People are a bit unsure. And it's a little bit up to us in a way on how we present and filter that data. And also highlight the errors that the other methods have. If you have a baited underwater survey, you're also dealing with relative abundances. It's also capped. So it's kind of like similar compared to eDNA, and it's another tool in the toolbox. So it's a really difficult question, but let me think about it a little bit more. And next, I can send you an email with some of our thoughts that we had with our policy people who are having also internally at Parks Australia, kind of like butting heads. So it's, I think it's more grassroots sometimes. It's kind of like having the empowerment from the rangers on the ground. What we have seen, if they're keen, they will advocate it up as well.
Yeah, I mean, I think, okay, just to paraphrase your answer, I think you were saying it's incredibly difficult and you need to integrate it very carefully.
Yes, thank you.
No, I mean, it's just really interesting. I think you guys have really sort of made a little bit more progress on a national scale than a lot of other examples. So it's just sort of, it's good to see how we can move towards talking about amplicon sequence variants to policy and informing policy and so on. It's a very important discussion. But yeah. I think we'll leave it here for now. So I just want to thank everyone joining our first seminar series, and in particular, I want to thank Dr. Eric Raes for just wonderful first seminar, and we hope to see everyone joining and the audience growing for our seminar series as it moves forward. So thank you once again, Eric.
Thank you very much.
Thank you. Thank you all.
Bye.
Bye-bye.
Bye, everyone. Thank you.